Fetal MRI, in vivo fetal MRI, I have to say it's one of my core research interests that I've been working on for a very long time, maybe around 15 years. And I will tell you about motion correction, different modalities, specifically structural and diffusion in this talk for the brain, and some applications that we have been working on recently. All right, so first I would like to introduce my patients. So this is what we deal with in fetal MRI. So for the first time ever, I put in the bracket in vivo because I always assume it's in vivo, but clearly not today. We have a postmortem topic as well. So all my work will be on in vivo MRI, and obviously you can see what kind of problems we have with in vivo MRI. I'm just wondering whether I have a pointer. Okay, good. So you see, this is a very young fetus, so it's moving a lot. The head can go quite a distance, which is difficult for motion correction. We have a mom that is breathing as well. It's moving the fetus up and down. Then for the resolution, we are imaging this tiny brain, but MRI has to have a large field of view because all mother is in the image. So we have a really difficult conditions for imaging. This baby is a little older, so it's a bit bigger brain and doesn't move as much. So they are a little bit easier to reconstruct. All right, so of course, ultrasound is the standard modality for imaging fetus, and just for comparison, this is the type of data that you get if you do 3D ultrasound. This comes from my work in Oxford. But obviously, we all want to work with MRI, and the reason is clear because the image quality we get is really good. We have exquisite soft tissue contrast, so we have insights into in utero development of the brain as a result. And also, we have versatility to visualize various tissue properties, and I will talk specifically about diffusion MRI in fetal brain today. So, outline of the talk. So I will start with T2-weighted structural MRI of the fetal brain, motion correction, super resolution reconstruction, and AI for automated and robust reconstruction. So the first two topics are very old, originating from 20 years ago. So it's just quick recap what many people already know, and some might be even expert on. Then I'll move to diffusion MRI, and I will describe the extra problems we have. So distortion correction and reconstruction of diffusion signal on a sphere, and then how we get the fiber orientations from it. And finally, I want to describe some studies we've done recently on fetal brain development. So let's start with structural. So this is the classic stuff. So because the baby is moving all the time, we cannot have a 3D sequence because 3D sequence just takes far too long. So rule of thumb is that the acquisitions should be under 1 second, otherwise it's too corrupted by motion. So only thing we have left is to do single shot imaging of 2D slices. And for structural imaging, we have haste imaging. So we acquire several stacks of single shot slices in different orientations. Then we start with volumetric registration. We select the best stack, do volume to volume registration to align all the stacks together. And then we do the first super resolution reconstruction, from which we get some not very good quality blurry target, but it's good enough to align each slice separately to improve the alignment. And we reiterate this process. Currently, we are doing just 3 iterations, and that's enough for the method to converge. So you see here a little bit through plane direction. This is kind of messy data that we get, and after reconstruction, we can have nice 3D volume. So a brief recap on super resolution. So basically, why super resolution is so successful in fetal MRI is because it models the forward model of acquisition to simulate the slice acquisition, and then we solve the inverse problem. So it does describe to an extent the actual process of getting the slice. And specifically in structural fetal, we tend to have 6 slices to get enough SNR. So we have a anisotropic blurring of the data, and this super resolution technique tries to account for it. So this is how it works. So we've done some registration. So we have motion parameters. So for each slice, we know where the fetal head should be in the scanner. So if I have some intermediate result for my 3D reconstruction, I can orient this 3D brain in the scanner. So I know where it would have been at the time that the slice got acquired. Then we apply the point spread function and we simulate the slice. And in my software, it's a 3D Gaussian, which is kind of a decent approximation of the point spread function, but it's not 100% accurate. And then we solve the inverse problem. It's maybe the most simple to understand when we use gradient descent. So the idea is that we compare simulated slice with acquired slice, and of course that will have some error, and we want to update the volume so that they fit better. So we start with some scatter data interpolation, which is like Gaussian weighted reconstruction. We initialize the volume, simulate the slice, calculate the error, and update the volume with gradient descent or a conjugate gradient descent equation. So the full algorithm that we are using corrects motion between slices, rejects corrupted and misaligned slices, reconstructs volume from scattered slices, applies regularization to the volume to reduce noise, and corrects for MR intensity artifacts. And here you can see how the reconstructed volume compares to acquired slice. So this is like slice from the volume that exactly corresponds to the acquired slice. And we could say that we do have a little bit better resolution, even though I don't know whether you can say that we really increase the resolution, but we certainly dealt with some blurring through plane. So the data look quite nice after reconstruction. So we've been doing that for many years, but what always has been a problem is when there is major motion, and this does happen, especially in early gestation. At later gestation can happen as well. The baby cannot move so much, but still able to turn the head 90 degrees. And if that happens, then classical registrations will always fail. So we get this kind of really bad result if we had this amount of motion. So what we've done, we worked on a deep learning solution. So our solution is not like fully deep learning reconstructions, which are now popular, and I know people are working on those. But we just have a deep learning as a support for our SVR, and that works really well. So this is work of my postdoc, Alena Us. So what we use deep learning for is for localization of the fetal brain. So you can see here, we also do that for trunk. So that way we know where the brain is and we do the masking. And then the strategy she came up with that she trained a network to segment the femur structures, not just the whole head. And based on these structures, it's then possible to reorient to the standard space. And so all the stacks are reoriented so that the brains nicely fit on top of each other. And then we can use the standard SVR slice to volume reconstruction, and it works well. We compared with NESWAR actually, and we are doing almost exactly the same as NESWAR with this type of approach. So this approach really just works based on segmentation and units. So it's quite simple, but very effective. So that's all about structural MRI. So now I want to move to diffusion MRI. So the extra problem, as I said, we have distortion And then also we have a multidimensional signal, so basically signal on a sphere. So we need to do something about that during reconstruction. So here I have all the challenges mentioned in diffusion. It's motion, distortion, intensity artifacts, then scatter data, interpolation, and then finally we need to extract fiber directions from reconstructed diffusion signal. So motion and intensity matching are the same as with structural, so I will just focus on what's specific to diffusion. And I will start with distortion correction. So this is kind of interesting because we have this geometric distortion, and in diffusion it's not actually in 3D, it's only in one direction, which is the phase encoding direction. So you can see in this direction it's all fine, even though there are the slices, but here we have this kind of weird shape, and that comes from the bubble in maternal gut. So, okay, so actually in terms of fetal brain, there are not any sources of distortion because it's all lots of water. We get distortion from the where we have air and tissue interface primarily, and that's maternal gut has that. And here the bubble was very near the fetal brain, so you can see it nicely here. This is B0 field map acquired, so we can see where the distortion is coming from. Okay, so because it doesn't come from fetal head, it doesn't move with fetal head, and this is important to realize because it's not the same as in neonates, because in neonates the air is all around the head, or in adults, and also in the ears, and that's where the distortion comes from. In fetus, it's all water, so it comes from outside, so it behaves differently. So it will not be moving with fetal head. It will be smooth, and it obeys Laplace equation. What it can move with, though, is with maternal breathing. So what do we do? So the solution that I developed a while ago was to jointly estimate motion and distortion using image registration. So for motion, we have rigid slice to volume registration, and for distortion, we have deformable volume to volume registration, because you can imagine that the distortion is in the scanner space, not in anatomical space of the fetal head. And but we constrain this registration to be only in phase encoding direction, so the control points only move in one direction. And we have also Laplacian constraint to make it more physiologically plausible. So here is how it works. So fortunately, we have already structural MRI that is not distorted, so we can use it as a target volume. So we have motion parameters as well from the registration. So we just again position the head in the scanner space or world coordinate system correctly. Then we simulate the acquisition of the slice. And then we can apply the registration in phase encoding direction only to undistort the distorted slice. And we have Laplacian constraint on deformation field, and you can see here how important it is, because we get lots of artifacts if we don't constrain it properly. And here you can see it's nice and smooth as it should be. This is acquired field map. It has a bit of artifacts as well, but we are much closer to acquire field map with the constraints that are based on the physics. So some results. So you see here that was what the bubble cost, and we nicely put it back. It's now fits with the structural data. And in this method, we apply it then to the diffusion. So this was done on B0, and we can also apply that to diffusion sensitized scans. So DHCP is not using this method. They are using acquisition-based method. It's I would say a little bit more accurate, but it requires a custom-made sequence. So it's not very easy to reproduce unless you have MR physics people implementing the sequence for you, but they are getting it from the phase of the gradient echo scans, which needed to be implemented. So that is, I would recommend to do that if you can, but if you can't, this method is always available. So a final bit of this, how do we reconstruct a diffusion signal? So what we need to realize that diffusion MRI signal is a function of spatial location and then sensitization gradient. And for each spatial location, which is a voxel, we have a function on a sphere. So I work with Donald Tournier on this, and of course that means that all will be spherical harmonics. Basically, this is spherical harmonics basis function, and we use that as a parametrization for the diffusion signal. So we have linear combination of these basis functions, which gives us these kind of little blobs here, which are diffusion signal. So I have to say it looks like blobs because this is diffusion signal, not the directions. They need to be converted to direction. I will talk about that in a little bit. So it's not on the grid, obviously, due to random motion. So we have scattered spatial location, just like with structural data, but also gradient directions will be scattered because every time we rotate the slice, we change the gradient direction. So we need to rotate it, and then we don't get it on a grid that we defined. So we need to reconstruct scattered data both in space and in the and on the sphere. But that's actually not such a big issue because we will do the same approach. We start with the forward model, which is always much easier to do how the data is acquired and reconstructed back. So let's say this is like some kind of latest estimate of the diffusion signal on the sphere in 3D, nicely constructed. So you start with something very simple at the beginning and update it. Then we will first apply the, I went one too far, the sensitization gradient, and then we get what the volume would look like with that gradient. So then we have a volume, and then we can apply exactly the same approach as before. We use motion parameters to position it correctly in the world coordinate system, apply point spread function, and we get the simulated slice, and we can compare to acquired slice and again use some iterative method to recover the volume like that. So gradient descent or conjugate gradient descent. So when we finally have our signal, so we've done this step, now we need to finally convert it to gradient to the directions of white matter, not just white matter, of the fibers. So to do that, we actually can use the same methods as they are for adults because now we are nicely on a grid. So we tidied up the data. So we just use a matrix for it. So here is a schematic. This is if you have just one bundle of one direction, then diffusion signal will be actually perpendicular to that bundle, right? Because where there is diffusion, we will lose the signal. So it looks like a disk like that. And then what we need to do is constrain spherical deconvolution, which we use matrix 3 for that. So from this kind of disk, we get a fiber orientation that is perpendicular to that. And of course, you can get crossing fibers as well with that. So this is the final result of the deconvolution. And here are some reconstructions from DHCP data for different ages. So the method works both for DHCP and standard acquisition, all the different gestational ages, and we can find crossing fibers as well. Then limitations. So for my technique that I presented is a single shell technique. We have for DHCP a multi-shell extension that is called shard that you maybe heard about. And yeah, if you are interested in my software, it's all available. You can email me for support. Now that's it for the reconstruction, and I have final topic, which is brain development, and I find this really exciting. This is our very recent work that we've been doing with DHCP data primarily, but slowly we're expanding beyond DHCP as well. So one of the things that we've been working on for now maybe 3 years is segmentation of fetal transient layers. We are really interested in those. So we have a trained convolutional neural network. It's an attention unit that was done by my PhD student, Helena. And we have cortical plate, subplate, fetal white matter, periventricular white matter. So we have 3 segments, C1, C2, and C5, and C4, and ventricular zone that is separated to ganglionic eminence is separate. So it's we have 2 PP papers published on that, but we are in preparation a manuscript that we plan to submit soon. We also get lots of help from Abby Gardner, who did the segmentations for us according to histological atlas in the spatiotemporal atlas space. So I love this 3D visualization because this blue surface or blue label, that's the subplate, okay? That's not cortex, that's subplate. And you can see at the beginning, it's completely continuous. And then as the folds deepen, like the sulci deepen, the subplate is disappearing. So every time sulci goes in, the subplate disappears. And you can see these holes appearing where the sulci are appearing. And at 36 weeks, it's already quite discontinuous. And as it goes with neonatal development, you will just have remnants on it. So it will continue disappearing. And here in yellow, I have 3 periventricular white matter segments. Helena just plotted for me the volume. So these are absolute volumes. So here you see what happens with volume of cortical plate. It's of course increasing, but is actually accelerating. While subplate is also increasing, but is actually slowing down and it's plateauing the volume towards the end of our age range. And just an example here, this is C2, C5. So I think that's this big arch. So it's first accelerating and then it starts stagnating because, of course, subplate and periventricular white matter are going to disappear after birth. So that's why it makes sense it's slowing down while cortex is still going to grow. So we started looking into what we can see in different anomalies, how these layers develop. And the data we had available were ventriculomegaly and Down syndrome. And we see in ventriculomegaly that ventricular zone has larger volume, which is kind of what was expected. We were happy to find it. And it's, I think these data were isolated left ventriculomegaly, and we still see larger ventricular zone both in the left and right hemisphere. For Down syndrome as well, the brain development is quite different. So we wanted to see what is different. And the most significant feature that came up was that the subplate volume was smaller. So this is still kind of a bit preliminary work, but I think for these 2 things, the results are quite strong. Then next, we try to look at microstructure of fetal transient layers, and the problem is we don't have diffusion data for all these different cohorts necessarily, so we mostly had enough data to look at normal development. So what we've done, we took DHCP reconstructed data and then took FA and MD, and we aligned it with structural, and there we have the segmentation. So we were able to calculate the average FA and MD per transient layer region. And this is MD, mean diffusivity. And what I found really interesting that we see mean diffusivity in general going up and then going down again, and it peaks around 28 weeks in general. So of course, mean diffusivity is like a surrogate for water content. So it seems like the water content in the brain is increasing and then it starts decreasing as it matures. That's how I interpret it, even though I'm not an expert on it. And we see this peak around 28 weeks. But if you look at some other zones, especially periventricular white matter, that's the bright bit of white matter, it seemed to peak a bit later, around 32 weeks. So this was quite interesting. That's what we saw in our DHCP data. Also, I think this is not transient layer, this is cortical plate, but I find it interesting because actually we see both mean diffusivity and fractional anisotropy decreasing in cortex, and it's kind of also like like they shouldn't be taken as an opposite measures because they don't directly relate to each other. So we know that we have this radial orientation in the cortex, so the fibers go perpendicularly to cortex in early gestation, but this is slowly disappearing as we go up with age. So it's much less coherent in the cortex at 32 weeks, and it become even less coherent later on. So that's why we see FA going down, because we're getting more orientation in the cortex with the maturation. But on the other hand, MD is going down as well, and I think it's probably because cell density increases. Again, I'm not an expert on it, but that would make sense. So actually, we see two different processes that are separate from each other with these microstructural measures. This is something very preliminary. We wanted to do to see what the microstructure does in ventriculomegaly, but we had only 6 subjects from DHCP because the other cohort didn't have diffusion data. So this preliminary data show that there might be some differences. We saw lower MD and we saw in cortex and higher in ventricular zone. So I don't know yet whether this will get like confirmed, but it's something interesting to look at. And final bit I want to tell you about is that we started working with Emma Robinson on cortical surfaces and looking at cortical folding. So my postdoc Irina, she's just finished a technique that fits the cortical surface to fetal data directly. So classical pipeline like DHCP pipeline would go, first we get the segmentation, then we fit the cortical surface, and then we would align these surfaces to the template to do analysis. What we realize is that because the cortical surface fitting method that we are using actually deforms a template to a subject, we already get the registration to the template. So we said, why don't we unify everything in one framework? So basically we get a fetal data set, select age match template, and then fit the surface. And basically we straight away have vertex to vertex correspondences that are anatomically meaningful. And the reason why that works is because of how we designed the architecture during training. So we have pseudo ground truth surfaces, which are from DHCP, from classical method, and we have images, so for supervised training. Then we use this network called COTAN that estimates the velocity field to estimate the deformation of initial surface to the subject, and for us is a weekly template. And then we predict the focal depth map from the predicted surface, and also we have focal depth map in the template, and we also ask that these are aligned. So we use just a normalized cross correlation during training. And then basically we have always, unlike in DHCP surfaces, we always have the same number of vertices, and they really do correspond between templates and between subjects because of how we design it. So we can do vertex-wise analysis straight away after surface fitting. And I don't yet have this kind of results because this work is really new. We are just going to submit to middle on Friday. So here you can see so-called depth maps for different ages, and this is mean curvature, and these are some whole brain measures like cortical thickness, so-called depth, curvature, and surface area. And that's all I wanted to tell you, just some thoughts about future work. So what is really high on my list of priorities, I would like to work on improving the diffusion reconstruction with deep learning, because there are lots of problems with diffusion data still that are unsolved. We would like to look at development and maturation of white matter tracts. We haven't done that yet. Oh, sorry, this is something. Oh no, it's just for me. You see it. Then I think it's really interesting to link the emergence of cortical folds to development of transient layers. Surely there must be some relationship between their shape and microstructure. And finally, we would like to look at altered brain development in high risk of autism and high risk of preterm birth. And we have diffusion data for that, but they haven't been processed yet. And that's all. Thank you very much for your attention. Oops, I just have some special thanks here on this slide.